Okay, we're rolling. Right, so week 9, as I said before, today is going to be pretty much focused on micro, so bacteria, viruses, fungi, the different infections you can have, the different ways that your body responds to those infections and cell damage, and of course the ways that we can, as doctors, seek to ameliorate them. So the very first thing we're going to talk about today are bacteria and viruses. Right, so we look at prokaryotes versus eukaryotes in the context of bacteria. So as you guys will know by now, viruses are acellular organisms, but bacteria are very much cellular. So they have similar chemical foundations, but they're structurally distinct. That's sort of a word salad. What it actually means is that we use the same amino acids, we use the same biomacromolecules, we use the same genetic code. All of that stuff is identical between prokaryotes and eukaryotes. Now, the difference is, of course, prokaryotes are smaller. They can only be unicellular, whereas eukaryotes can be unicellular or multicellular. Prokaryotes divide by binary fission, eukaryotes by mitosis, and prokaryotes don't have organelles or a nucleus. Now, what they do have on eukaryotes is peptidoglycan, a very complex cell wall, which we're going to talk a bit more about in just a second. And that's actually going to be quite important because it's going to be how we target bacteria, because that's a unique feature of bacteria and prokaryotes in general. We also find them in one of 3 major shapes. So you'll notice this is in the name for the vast majority of bacteria. So you'll see like Streptococcus staphylococcus. What that means is that it's hanging out as this little ball-shaped thing. Bacilli means it's sort of a pill shape. And spiral means it's sort of this like almost little like pasta shape, if you ever had those twisted pasta things. All right, now let's talk about the cell wall. So there are cell walls present in some eukaryotic cells, typically those of fungi or plants, but the bacterial cell wall is unique. And that's because it contains a complex carbohydrate known as peptidoglycan. So the Gram stain is a test that we use to differentiate between types of bacteria, and it's based on this peptidoglycan. So all the bacteria have peptidoglycan, but Gram-positive bacteria have a very thick peptidoglycan layer, which forms the entire outer surface of the bacteria. On the other hand, Gram-negative bacteria have a thinner peptidoglycan layer, which is not on the outside of the cell. They have something called lipopolysaccharide on the outside of the cell instead. So when we stain them with the Gram stain, it's a whole process. You don't need to understand everything about it. But basically, Gram-positive bacteria have a darker purple stain. Gram-negative bacteria have a pinkish red stain. Again, you don't really need to know the workings of it, just the fact that Gram-positive means purple, Gram-negative means pinkish red. So we also have certain bacteria which just can't be Gram stained. So they don't give us a positive or a negative because they have an extra waxy sort of lipid layer. It actually the name Mycobacterium actually comes from that substance called mycolic acid. And so we need to use what we call an acid-fast stain on those bacteria instead of the normal Gram stain that we would typically use. All right, so bacteria can have a variety of exterior structures, and some of these are just useful in general terms. Some of them are specifically a bit of a hassle for our immune system to deal with. So we've got first the idea of a flagella. So it's like a big whip that comes out from the back of the bacteria. It's made of flagellin, and it rotates like a propeller. So it's effectively like a paddle or something for the bacteria to use. Obviously, the bacteria needs to be in fluid for this. So this doesn't work for bacteria when they're in just like a dry environment or something. They need to be in a fluid environment where they can use this to act against gravity or move around it, that sort of thing. You don't need to only have one. Some bacteria only have one. The micrograph we've got on the right there shows a bacteria with multiple flagella. We've also got something called a capsule. So this is usually made out of polysaccharides, so sugars. And it has a whole bunch of functions. They basically all come to being protection or adhesion. So it both protects the bacteria in a general sense from the environment, so desiccation. It also makes it harder for phagocytes to actually eat the bacteria. So that's a big problem for our immune system because obviously phagocytes are the primary way we get rid of bacteria. And it can also help it sort of hide a little bit because it can often be chemically similar to your host cell sugars. So when you're trying to, when your immune system is trying to differentiate between the self cells and the non-self cells, it can often be fooled a little bit by these capsules because they're too similar. And it also finally aids in adhesion. So these guys like to stick to different parts of your body in order to stay in place. And this actually helps them with that, which is not good for you. One more thing, look at how big those capsules are. So proportionally, we want to be remembering that these are like really, really big structures. It's not a thin layer. It's a very, very thick one compared to the size of the cell. All right, we also have more adhesion molecules, and these ones are specifically adhesion, which are called pili or fimbriae. So you can see there on the right micrograph, there's a whole bunch of sort of hairs that come out from the cell surface, and they help the bacteria stick to pretty much anything. So random surfaces in the environment, the host cells of the organism that it's infecting, mucosal membranes, so in your your lungs and things like that, in your respiratory tract, or other bacteria. And probably the biggest problem with these, or the biggest danger to us in a clinical perspective, is that it allows something called a biofilm to form, which basically means a huge massive bacteria and all of it's connected to other bacteria. So it forms a big sort of continuous surface. And it's a lot harder to get rid of a biofilm than it is to get rid of scattered bacteria, because the bacteria sort of give each other a level of protection and structure. So we, in terms of like disease control, we spend a lot of time trying to make sure the biofilms don't develop. And we've also got spores here. So basically only certain bacteria produce spores. They're more common with fungal infections, but certain, like the ones that do produce spores are usually quite well known. So, Clostridium tetanus and Bacillus anthracus, so anthrax, they both make spores. You can only make one spore per bacterial cell, and it allows it to basically survive in an environment where it normally wouldn't survive, and then reactivate, come out of hibernation, as it were, later. All right. Now remember, we have a normal flora in our body. It is normal and good for you to have a whole bunch of bacteria on you. It actually forms part of your body's your first line of immune defense, is the bacteria that live on you and which don't cause infection. And that's most of microbes. So less than 1% of your microbes are actually responsible for disease. But that's not to say that natural flora is entirely risk-free, because there are certain bacteria, also other organisms, but we're looking at bacteria here, which can cause opportunistic infection. So basically they live around your body normally, and they're usually okay, but in certain conditions, usually when there's some way for them to get where they shouldn't be, they can cause an infection, and then they can become a danger to your body, obviously. So Staph aureus is a huge one. You see that all over the place. It's just a massive nuisance pathogen. Staph epidermis is also a big one. In your mouth in particular, Streptococcus viridans is one. And there are various forms of streptococci in your stomach. So quick note there, you remember coccus. So all of these are going to be little balls, tiny little sort of spherical shaped bacteria. There's a particular bacteria that I've highlighted there. So these are lactobacilli, and they're a particularly helpful mutualistic bacteria. They often make up a large part of the bacteria in the gut and the vagina. And the presence of a lot of this type of bacteria is generally protective against other infections. So you want to have more of the good bacteria and less of the bad bacteria. And so you'll often see a lot of probiotics, which include lactobacilli. That said, there are certain places where you don't want normal flora. So there's usually like your mouth, your gut, your upper respiratory tract, that sort of place. You want to have normal flora, that's normal. But there are certain parts of your body listed there which need to be sterile. So if there's bacteria there for any reason, that's a bad thing. Whereas in other places, only certain bacteria are going to be a problem. And that's a large part of opportunistic infection. So if something gets into the blood, if something gets down under the skin, that sort of thing. Basically things getting further in than they should. All right, these are the medically important bacteria. I'm not going to go through all of them now. Some of the important ones for you to just be familiar with, like you should be able to recognize the name, the Staphylococcus, your streptococci. These are just really common all the time. Clostridium perfringens, you'll learn about a little bit more. It does some cool stuff. Well, cool is a relative term. You have your gonorrhea and meningitis, which are responsible for the disease with the same names. And then you've got things like E. coli and salmonella. So you'll notice certain trends here. Generally, bacteria with similar properties live in relatively similar ways. So that's not to say that everyone in each category will have the same effect on the body and the same living style, but a lot of them can be found in similar places. Oh, one more thing. So one takeaway I do want you guys to take away from this is that we have certain things which are like the main ways we differentiate bacteria. So the gram stain is a huge one. It's just a really good way to differentiate between different bacteria because it gets to that structure of the cell wall, which is quite important. Then we've got these shapes. So here we're only looking at cocci and bacilli. We're not looking at any spiral ones, but they certainly do exist. And then we also have anaerobic versus aerobic bacteria. So those are all important distinctions which have implications for how we treat them and also how we classify them in the first place. All right. So viruses, as I said, they are not living. They're not actually organisms. They can't survive on their own. They need to reproduce by invading hosts. And they're usually specific to a certain type of host. So a certain animal, a certain plant, a certain fungi, a certain bacteria. Over the right here, we have two examples of a virus, and you can see the same sort of basic structure to them. Inside the that little sort of polyhedral shape, we have the protein capsid. Sorry, the protein capsid is the polyhedral shape. And then we've got a little viral genome in there, which is just a little bit of nucleic acid. In one of them on the left, bacteriophage, we've got a DNA bacteriophage, whereas on the right, we have a RNA influenza virus. Now, they may have an envelope of stolen cell membrane. So they can't make their own envelope because they're viruses, but they steal your cell membrane after sending viral proteins there to be embedded. And so you can see here immediately bacteriophages. So that's a type of virus which attacks bacteria specifically. And so they're specific to bacteria. We don't need to worry about these so much. In fact, we sometimes use them to treat bacteria. Influenza, on the other hand, we are not very happy about. All right, so we also need to classify viruses. And so we have a whole bunch of different qualities that we differentiate them on. So the genome, the genetic material of the virus, it's always a nucleic acid, but is it DNA or RNA? And is it single or double-stranded? You'll learn a little bit more about the distinctions here in semester 2. We also look at the size and the shape of the capsid. So that protein coat, it can be pretty big, it can be pretty small. It can be different sort of shapes. And we can also look at the symmetry of the capsid. So sort of the classic one is this icosahedral shape, which is basically like a big polygon sort of shape. Whereas the helical structure here is obviously a lot longer and bigger. So you can see there's quite a big difference in shape there. The presence of an envelope. So is there an envelope or not? It's true for certain viruses, it's not true for other ones. The hosts, so different viruses are specialized to different hosts. We've got lots of different flu viruses, a lot of them are specific to animals. And finally, the disease that comes from them. So that can be important when we're talking about how we classify them. Now I want to stress, we've got sort of like a family genus structure here. That doesn't mean that this fits into the like classification of everything else. So we've got the whole big universal classification system for all living things. These are outside of that because they're not living things. When we talk about viral families, genuses, viruses, and strains, they don't fit into any kingdoms or phylums or anything. They just stand on their own. All right, so how do viruses replicate? We said they need to hijack a cell, but it's pretty simple in the end. So they attach to the cell surface. They've got some combination of receptors and markers which allows them to do that. Then they trick the cell into letting them in. So there's various mechanisms they can use that, again, probably next semester. They replicate, so they basically hijack the cell's ribosomes, the cell's various construction facilities inside there to make more viral material. And then they assemble those viruses. So they make those capsids, they make those nucleic acids, gives them the structure to form a new virus. And then finally they release, so they leave the cell. Now, if there's an envelope, we have something called budding. You can see here, this is the on like the right side of the release thing in the diagram, and we can see budding, in which they sort of the opposite of exocytosis, in which they take off a whole bunch of cell membrane. Now, if there's no envelope, they just lyse the cell. So this is like guaranteed to kill the cell because it's just splitting open the cell membrane and letting the viruses out. So the actual mechanism by which it leaves the cell is going to depend on whether it is an enveloped virus or not. All right, so this is a whole bunch of big words. They're pretty simple. The diagram on the right is quite helpful. So abortive infection means nothing happened. An acute infection means that you you've got viruses replicating inside you. Typically, these are ones which damage the host cells, because if that is happening, if your host cells are damaged or dying, then either the virus wins or you win. And so we have, like right at the top there, common cold, typical one, your body will win, viruses replicate, and then eventually they die down again. There is, however, a risk of complications. So things like measles in particular, you can have a bit of viruses and then you think it's gone, and we have no really way of telling if it's still there. And then years later it can pop back up. Then we also have persistent infection. So this is usually, it has more subtle effects on host cells, because the ones with major effects, basically, they're too hard, they're too big to miss, basically. So you have a normal sort of chronic infection, which is just we have a whole bunch of bacteria all the time, and we have shedding of the virus. So it's like, you constantly have viral particles and viruses being released, which makes you quite infectious. We also have chronic infection. So we have, again, the same sort of thing, except this one has a higher risk of having later disease episodes in which there actually becomes a significant impact on your body function. We have latent infection. So your viral genome stays in your cells, and then at some point it can flare back up if you're unlucky. And finally, we have transforming. So this one is probably the worst one for you because it causes cancer, basically. And things like HPV and Hep B are particularly risky for that. Don't worry about the last one in this diagram. That's for a prion, which you might learn about later, but is not a virus. Right. Medically important viruses, just know the names. Influenza and measles cause the diseases of the same name. Rotavirus is one of the important ones for gastro, which you'll probably be learning about in a ICL soon. And then these guys on the left just cause a whole bunch of nasty things. And again, so keep track of what we use to differentiate them. DNA versus RNA viruses is one of the big ones. All right, there's something in chat. Is that a question? All right, do we need to memorize the medically important bacteria? I wouldn't say you need to memorize. I think when you do more micro stuff, you'll sort of just absorb it. I'd say they'll talk about at some point like infections of various parts of the body, and they'll mention certain specific things which happen. So you'll want to like take note of the ones they highlight there. So they'll probably talk about things like Staph aureus, Staph epidermis, Strep viridans, Candida, which is a fungus, and various things like that. So the ones that they highlight, pay attention to those and learn those. And then this is a pretty good guide as well, the stuff we've got here for which bacteria are important. I'd say probably be familiar with what they do, because they might give you questions in which they ask you to link, a certain presentation or something with the responsible pathogen. All right. With that, I will hand it over to Marwan.
Thank you, James. For some reason, I can't share my screen. James, I think just the host can.
Yeah, gotcha. Okay.
Thank you. Can you see the screen? Right. So let's start by talking about the natural barriers and innate defenses. Actually, most of this content we already went through in week 6 in innate and adaptive immunity. I think this was done by James. So we'll go through some basic things just for review, and we went through some terms like inflammation, fever, and talk a little bit about natural flora. So let's start with some terminology. The disease can be defined as an interruption of normal structure or function of a normal human body, and it's usually defined with some associated symptoms or signs. This disease can caused by viruses, bacteria, fungi, yeast, and other microorganisms. Um, Immunity can be defined as the ability to resist infections, and we have two types of immunity, or two immune systems. We have innate immunity or innate immune system, which is an antigen non-specific immune system, and then we have adaptive immunity, which is antigen specific.
Can I interrupt you there real quick, Mohan? Do you have something across the bottom of your screen? Because the very bottom of the slide isn't visible on the screen share.
Just
There's still a big black bar.
Is it better now? Or?
Yeah, you want to try present again then? So like press the present button again, see if it works this time. Yeah, there we go, perfect.
Thank you, James. Thanks. So natural barriers are part of our innate immune system, like first and second lines of defense. There's some anatomical barriers, mechanical barriers, antimicrobial or chemical factors. We have cellular components, like white blood cells, and then we have the microbiota, or our normal flora, which we went through in detail in week 6. Now let's talk about inflammation. So inflammation can be considered as part of our innate immune system. So it's a non-specific response to injuries, pathogens, or irritants that occur in vascular tissue. It has 5 signs: redness, heat, swelling, pain, and loss of function. We also went through that in week 6, just as a reminder. And local inflammation actually is a good one, because it prevents the damage from spreading to another sites in our bodies. Then we have fever, which is a natural response to a number of illnesses that are often associated with inflammation. There are some signs for fever, like the temperature will be above 37, so it's the normal temperature. And it's usually associated with sweating, hyperventilation, and vasodilation in the skin. How does it work, or how can we consider fever as part of our innate immune system? Because fever increase our temperature above 37, it actually speeds up some immunological reactions that need high temperature to be speeded up. Also, some pathogens, like some types of bacteria, can't live in a temperature above 37. So by increasing the temperature, actually fever kills these pathogens. Finally, we'll talk about normal flora, which James went through just before. So normal flora is microorganisms that live on or in our body and cause no harm. Usually it's bacteria. There are 2 types of normal flora: resident flora, which stays in our body forever, and transient flora, which comes and go. And I don't know if we can consider it as adaptive, if you can call it adaptive flora, because we gain it after shortly after our birth, and it covers most of our parts in the body, as James said, but some parts like brain, blood, and cerebrospinal fluid, they have no normal flora at all. So if there was any presence of any microorganisms in these in the brain or in the blood or in this cerebrospinal fluid, this indicates that there is an infection to be treated. And how does it work? It prevent the colonization by potential pathogens. So we can consider as like compete other pathogens in these surfaces. Some of the specific normal flora functions, like in skin bacteria, it block off other bacteria from invading or other pathogens, and it produces fatty acids. For gut bacteria, it helps us to digest food. It causes other bacteria, and it can be considered as training ground for our immune system. Then we have the vaginal lactobacilli, which is an acid environment suppressor growth. So now we will go to second station, strategies for control the microorganisms. We've talked about bacteria, viruses, and fungi. Now we'll talk how can we control these microorganisms. There's something called the chain of infection, which is like steps that exist to make an infection spread. So by knowing how the infection spread, we can actually prevent the infection from happening. And this is very important to control the microorganisms from spreading. For example, we need to prevent infectious diseases. We can use these control strategies in some industries, like we need some types of bacteria to produce insulin. We might also need to control these microorganisms in some research in the lab. And finally, for hospital-acquired infections, how can we prevent hospital-acquired infections, which is called nosocomial infections. This is the chain of infection, and we'll go through each part. We've already went through the infectious agent, which involves these microorganisms or pathogens. We'll first go through the reservoirs. So the reservoirs, I don't know if I'm spelling that correctly. Reservoirs is where the agent survives or multiplies, where this microorganism lives. And we have three types of them. We have humans, animals, and inanimate objects like water or tools that we use every day. These microorganisms in humans can be present and they can cause just symptoms. So they can be symptomatic, like influenza, like where we can actually see the symptoms or signs of these microorganisms, or the humans can be just carriers for these diseases. For example, asymptomatic HIV in early stages. Then we have convalescent, for example, salmonella, and then we have chronic infections or chronic diseases like hepatitis B. Now we'll talk how these microorganisms move or how can they spread. So they spread in a number of ways. Firstly, direct contact and indirect contact. Direct contact, where it happened from directly from where it can spread directly from one host to another, and indirect contact, where it need to be exudated or secretion for another host. It can also spread through droplets, for example, influenza, SARS. We have right now COVID-19. And it can spread also through air, so airborne. For example, tuberculosis. It can also spread through inoculation, like if something bites you. This can be called inoculation. For example, insect bites. It can cause malaria or sharp injuries. It can cause hepatitis B and C. Also, it can move through vehicles, through water, food, or medical instruments. And finally, it can spread sexually or transplacental or in utero. And we can remember this for this torch. So T stands for toxoplasmosis, or for others. R for rubella, C for cytomegalovirus, CMV, and finally H for herpes. And now we'll talk about the portals of entry and exit, like where can these pathogens enter our bodies. So there are 6 types of portals: respiratory, conjunctiva, urogenital, gastrointestinal, skin, or placenta. And then the susceptible host. How can these pathogens or these microorganisms cause harm or cause the disease to the susceptible host? What are the factors that can cause this? So it can, like underlying diseases, can be through antibiotics, steroid immunosuppressive steroids, through surgeries, like in hospital, invasive devices. Also, young people and old people, their immune system considered to be weak, so they are highly susceptible to get these infections, and then immunocompromised. For example, people who are HIV positive, and then the malnutrition. So how can we control this microbial growth? We have 3 ways. We have cleaning, disinfection, and sterilization. Cleaning involves the removal of bacteria of any organic matter. Moist organic matter supports bacterial growth, like bacteria grow fastly in moist environment, so it should be removed immediately, and it must occur prior to any disinfecting or sterilizing procedure. Then you have the disinfection, which is like higher than cleaning. It involves the removal or killing of most viable microorganisms. For example, like skin disinfection, sanitization, or antiseptic. So it can be physical or chemical. And finally, we have the sterilization, which is the top way, or the best way to control microbial growth. So it's involved removal or killing of all viable microorganisms, including spores, and it can be physical or chemical. We'll talk about this in detail now. And there are some factors that influence microbial growth, as we mentioned before: moisture, temperature. Some microorganisms can grow in high temperature, osmotic pressure, pH, presence or absence of oxygen, and nutrient availability, and which level of control, like do we need cleaning or do we need disinfection or sterilization to clean this object. It depends on how high the risk can be this object for us. For example, if it can penetrate the skin, then we need sterilization. But if it may contact skin but not penetrate, so it can be considered as lower risk, and we just need disinfection or cleaning. So sterilization, there are a number of ways to sterilize things. Firstly, heat. So in other words, we cook the pathogen. We kill them by cooking them. So it is highly efficient. We have two types of heat: moist heat, which 121 degrees for 15 minutes, or 134 degrees for 3.5 minutes, done by a device called autoclave. It causes increase in pressure, increase the boiling point, which lastly produce higher heat, and it causes inactivation of proteins of these pathogens. Then the dry heat, which is 170 degrees for 1 hour or 160 degrees for 2 hours, and it causes inactivation of cell components by oxidation. Sorry. Then we have the radiation. So radiation actually damaging the DNA of these microorganisms. We have ultraviolet, so it's not very penetrating, and it's good for cleaning medical instruments, fluid, and air. Then we have ionizing, so it produces free radicals which break down the DNA, and it's good for heat-sensitive materials. Also, filtration, which we need which we use if there's no other way of sterilization can be used. If we can not use heat or radiation or chemical, and so it physically removes microorganisms from heat-sensitive liquids or gases. Finally, we have the chemical sterilization, and we use the chemical sterilization for materials that cannot be put under high heat. So one example of this is ethylene oxide gas, which is the most commonly used. And then we have the chemical disinfectants. We have chlorine, phenolics, iodine, chlorhexidine, and each one of these used for different purposes. And this depends, like disinfection and sterilization, depends on a number of factors, including number and type of microbes to be destroyed, purpose of treatment, does these devices that we need to remove the microbes from, are they considered as very high risk or others or very low risk? Then type of item, interaction with other active chemicals, toxicity, and finally cost. Now we'll talk about the classification of items or devices. How can we say that this device needs sterilization or disinfection or cleaning? So we have classified them into three. We have critical items, which are the objects that entering any sterile tissue, vascular system, or piercing mucous membrane. So these critical items, we need them to be fully free of any microorganisms, including spores. For example, surgical equipment. Then we have semi-critical items, which doesn't have to be to need sterilized. We just need to disinfection, high level of disinfection. So these objects come in contact to mucous membranes and non-intact skin. So we need them to be free of all microorganisms except these that does have spores, for example, flexible endoscopes. And finally, we have non-critical items, which can be cleaned through low level or intermediate level of disinfection. So these are the objects that come in contact with intact skin and not the mucous membrane, for example, the stethoscopes or bed pans. Now we'll talk about the nosocomial infections, or the infections that happen in the hospital due to some mistakes that happen during sterilization or disinfection procedures. So how can we prevent this infection from occurring? First, we can exclude the source of infection. By excluding the source, we know where they're coming from, and we can prevent them from spreading. We need to monitor sterilization, disinfection procedures, prevent transmission from infected patient to other patients, and implement walk restrictions. We need also to enhance host ability to resist infection. We need to strengthen the immune system by active immunization, passive immunization, prophylactic use of antibiotics. We need also to control of airborne transmission, protective isolation, and finally, aseptic behavior like hand washing, hand hygiene. And that's it for strategies of control. Now we will move to the fungal infection. So fungi or fungus eukaryotes that are obligate aerobes. They can be reproduce sexually or asexually. And one important feature of fungus that they have chitin cell wall with beta glucans and ergostriol. So it's like our cholesterol that we can find in the cell wall or in the plasma membrane, sorry. And we have three main types of fungi. We have yeast, molds, and dimorphic, which we'll talk about in detail. So the first type, yeast, they divide through budding and producing daughter cells. They are considered unicellular. And some examples of them, we have Candida, Cryptococcus, and pneumocytes. We'll talk about each one of them later and what are the possible diseases that they can cause. We also have molds, another type of fungi. They grow by extending hyphae to form mycelium. They are multicellular and they produce fruiting bodies or spores, which can be breathed in. So mouths usually cause allergies. And then we have dimorphic fungi, which exist in yeast and mold form. So molds at 25 degrees and yeast at 37 degrees. We have the Penicillium, Candida, and Sporothrix. So fungi can cause, some types of fungi can cause diseases through allergies by causing allergic reactions. They can also cause disease by producing toxins, and by finally by infection, by causing an infection, which can be cutaneous and subcutaneous or invasive, which we'll discuss now. So types of mycosis or fungal diseases. We have firstly this superficial mycosis or cutaneous. So this type of disease where fungi stay on the surface of the skin, hair or nails, like on the surfaces, and they require keratin, and they can cause skin to crack, leading to secondary infection. They can transmit from human to human contact, animal, or from soil. Fair, warm, humidity, and sweaty places. For example, we have tinea, which can also infect head, scalp, and body. It's causing the athlete's food, and it can be treated with azole antibiotics. Then we have the subcutaneous mycosis. They mostly occurs within inoculation, like for example, if wound from a rose thorn. One example of subcutaneous mycosis is Sporothrix shinki. So it's sustained after inoculation, and it can stay local in the site of infection or site of inoculation, or it can spread through lymphs, and it's usually treated with itraconazole and saturated potassium iodide. Then the last type of mycosis is systematic or deep, which forms internal organs, and it can be very dangerous. One example of mycosis is the Candida. So it can be part of normal flora opportunistic pathogen. It's very dangerous, so it have a very high mortality, 40 to 60%. It's very resistant to treatment. We have these examples here, specific examples, and it can be diagnosed through culture or PCR. Then we have the Cryptococcus, which is small budding yeast that has an outer capsule. It also have a very high mortality rate, higher than 50%, and it can be diagnosed through Indian ink stain or through culture. Also, another example of deep mycosis, we have Pneumocystis. So it's a yeast-like fungi. It infects usually people with immunocompromised, who have immunocompromised system. For example, people with HIV. It can cause pneumocystis, pneumonia, and it can be transmitted from person to person through air. Some of its symptoms: shortness of breath, dry cough, fever, weight loss, and other pneumonia symptoms like chills. It can be diagnosed through PCR or microscope. Finally, we have Aspergillus. So it's not normal flora, and it typically infects lungs and sometimes brain. So it's also very dangerous, and it can be diagnosed through microscopy or culture from the sputum. And that's it.
Alrighty. I'll share my screen if that works.
Where is it? You next or me next?
Oh, good question.
I think I go diagnosis and then you go.
Oh yeah. My bad. My bad.
No worries, he's busy. Okay, give me 1 second, folks. Okay. I'm assuming we can all see that. Alrighty, so let's go for diagnosis real quick. So this isn't going to be super in-depth. We're going to focus basically in on the most common and most important methods of diagnosing a pathogen. So we're going to start off with the really big ones, which are the bacterial methods. So these are, there'll be a really good flow chart here to explain how these all fit together and how we use them to sort of slowly narrow down what pathogen is the causative agent of an infection. But these are the ones which are really important distinctions because they help us differentiate very efficiently between bacteria. So we've already talked about how the gram stain works. It's pretty simple. We can test if a bacteria has a thick peptidoglycan or a thin one. There's also shape. We talked about that as well. Cocci versus bacilli versus spiral. Sphere versus pill versus twisted pasta shape. There's something called catalase as well. So this is basically just we pour H2O2 onto the cell or the bacterial sample, and then we see if we get hydrogen gas out of it. Oxygen gas out of it. And so that's basically just testing if we have catalase, which is something that you find in your body, but it is not found in all bacteria. So we check if there's catalase there. And so the presence of the gas is considered a catalase positive test. Coagulase, so we've got, we also tested for an enzyme here which causes clumping, and this is regarded as a positive test if we get coagulase. So we also want to keep in mind which outcome is considered a positive test. And with the gram stain, obviously that's the purple color for the thicker cell wall. There's also hemolysis. So this one is sort of a weird one, but once you understand it, it'll be pretty easy to remember. So it's basically testing if the bacteria breaks down red blood cells. So we use a particular type of agar called horse blood agar, which is exactly what it sounds like. It's agar with horse blood on it. And so we'll put the bacteria there. It's agar, so it has nutrients. It's able to live and grow. And then we test we see what happens to the blood cells around it. It's a little bit confusing because it doesn't just go alpha, beta, gamma like you would expect. Instead, alpha is the partial hemolysis. So you see there in the middle image, there's sort of like a kind of gone but not completely gone substance there. You can see the bacterial colonies in a big sort of swish across the surface, and then the cells around them are not doing the best, but they're not completely gone. Now, beta hemolysis, we do have them completely gone. So they like completely destroy any red blood cells in the vicinity. And then we have big clear zones around the bacteria where nothing, no blood cells are remaining. Gamma hemolysis, you might see a little bit of discoloration where the bacteria are because the bacteria are there, but you don't see any hemolysis. All right, now this is the flowchart I was talking about, which is a really good illustration of how we can sort of go through diagnosis. And this is, for most diagnostic methods, this is the basic idea. This is what we're trying to do. We use the biggest methods first, so the things that help us narrow it down the most. And then as we slowly progress, we get a better and better idea of what pathogen might be responsible. And then from there, we can slowly narrow it down until there's only one or two that we think it might be. And they'll help inform the actual treatment that we give the patient. So you can see here, we start with the gram stain. We take a sample, we check what shape it is, then we go to catalase, coagulase, hemolysis, all of these tests. This is another good list of important bacteria. So somebody asked before, do we need to memorize important bacteria? I'd say all of these except for maybe S. agalactiae, I'm probably pronouncing that wrong. We didn't talk too much about that, but the rest of them all come up and are all quite relevant for you. All right, and we do also have some more general techniques. So obviously the stuff I mentioned is first of all just sort of very general big stuff, and also is only useful for bacteria. So sometimes we need to do other things. So when we talk about microscopy, we have two types, which you probably talked about in histology earlier in the semester. So light microscopy, you literally shine some light onto a sample and then you check what's there. So that's good for larger microorganisms because we can get down to the right magnification for them. It's not good for small microorganisms because you just can't see them at the scale of light microscopy. So instead we use electromagnetic microscopy. So we're talking about electron microscopes here. And then that allows us to see much smaller structures. So your viruses, as we know, they're way smaller than bacteria or human cells. And so we need EM microscopy if you want to actually see them. We can also do culture. So I've said bacterial culture, that should be both bacterial and fungal culture because we can do it to both of them. We basically just provide, something like agar, which gives it a lot of nutrients. We incubate it, we make it very nice, and that'll help it grow a colony. And then once we've got a colony, we can just test that and see what's in there to figure out what's actually wrong with that patient. Serology is when we identify antibodies. So this is actually testing for the body's response, and there'll be a few more techniques that do the same thing. But it's basically predicated on your body has had an adaptive immune response, and then we test for that. Ribotyping is when we take a DNA sample out of something like a virus or a bacteria, and then we use DNA comparative analysis to figure out what the DNA came from. And yeah, so the polymerase chain reaction is sort of the same thing, or it's a technology which we can use for things like ribotyping. And we talked about this earlier in the year, I think during biotechnology. It's basically you have small DNA sample, you go through PCR, and you have a lot of DNA samples. MALDI-TOF is a specific type of mass spectrometry. You don't need to be too well known on how it works, but basically the upshot is that it helps us identify pathogenic proteins. And the last two methods here are both, again, testing for your own body's response as opposed to the pathogen itself. So ELISA, enzyme-linked immunosorbent assays, is basically, we have a whole bunch of antigens there, and then we expose a sample to them and we see which ones bind where, which basically tests for a whole bunch of different potential pathogens. And if your body has a reaction to one of them, then we can see that. And then precipitation and agglutination, if you remember our presentations on the immune system, you'll remember that these are different things that antibodies can potentially do. So agglutination is slightly more sensitive. You're more likely to have actually like a correct positive result, a true positive with agglutination, but both of them are quite useful. All right, short and sharp, I'll hand it off to Raymond there to talk about hemostasis and coagulation.
Awesome. All right, let me try and get these slides going or sharing screen. Try again. Yep. All right, let's try this. Oh, nope. Check screens. Look, that works. Awesome. Oh, let me go back. So we'll be talking about hemostasis and coagulation. So as we know, homeostasis is the process in which the body keeps itself in a optimal working condition. Hemostasis is basically that, but for the blood. And to keep blood in a working condition or optimal range, you want it to be kept inside your body and not flowing outside of it. So basically, hemostasis is the physiological process to prevent and stop bleeding. And the major factors involved are platelets, fibroblasts, and coagulation proteins. And basically the basic process in which a clot forms and then dissolves and disappears is that an injury occurs, a platelet plug forms, and then this is then reinforced with fibrin. And then once the wound has healed, the clot is dissolved by fibrinolysis, and then we return to normal function. So first and foremost, we have primary hemolysis, homeostasis, which is basically the formation of the primary platelet plug. And the sequence in which this occurs is that the endothelium cells of your blood vessels are damaged in such a way that the ECM is exposed, which is rich of collagen and this factor called von Willebrand factor, which is produced by your fibroblast. And that's why they're important in this. And these collagen and von Willebrand factors, which are proteins, they basically bind to platelets, which are flowing in your bloodstream, which are created by your megakaryocytes. And these cause them to go from an inactive state to an active state. And during that process, it results in the release of serotonin, which causes a vascular spasm, therefore resulting in the constriction of the blood vessel to reduce the amount of blood going through the area to prevent or reduce bleeding, as well as the release of ADP and thromboxane alpha 2. And these are basically platelet signaling molecules that cause for more platelets to aggregate in the area in basically a positive feedback loop. So the more of the platelets that are activated, the more that come and are recruited, resulting in more platelets. And then these are also, the activation of these also results in cross bridges forming by fibrinogen cross bridges forming the primary plug. And this forms the site of coagulation or secondary hemolysis. So basically at this point right now, we have, as an analogy, we have a hole and we've just plugged it up with maybe some paper mache, and it's kind of iffy and probably going to fall apart if we don't do anything about it. And what do we do about it? That is the coagulation part, which is the secondary hemostasis, which is when we reinforce this clot with fibrin. So you can think of it like, if you have concrete by itself, it's kind of useless, but if you reinforce it with rebar, it becomes a lot stronger, and that is secondary hemostasis. And basically the primary plug provides a template for where the concentration of coagulation factors can form, causing reinforcements. And this comes in the form of a cascade known as the conjugation cascade. And it's known as a cascade because it results in a huge or multiple different coagulation factors acting upon each other, resulting in factor A affecting factor B, factor C, so on and so forth, until we get a stable clot known as the coagulation cascade. Basically, in this cascade, we have coagulation factors, and these are basically and they're named as coagulation factor 2, 7, 9, 10, 13, and so on and so forth. You don't actually have to know the exact names because it's just too much information. You just need to know that they exist and they are produced by the liver and activated during secondary hemostasis. And some of these factors require vitamin K. So if you have a deficiency in that, you might see patients that have problems with clotting. Also, if the patient has liver damage and they're unable to produce clotting factors in general, they will also have problems clotting. And in terms of the cascade, there are two pathways, but they all converge into one common pathway, and they're the intrinsic, which is initiated by when blood vessels are damaged, and the extrinsic, which occurs when tissue is damaged. But they all converge into the common pathway as they all encode for an enzyme that it starts up the common pathway, which is known as coagulation factor 10. Here is an image of that coagulation cascade. And you can see over here, there is, as you go from the intrinsic pathway and the extrinsic pathway, they all end up producing coagulation factor 10, as shown in the Roman numerals. If you're confused by this image, the A's just means an activated form. So all of these are just floating in your blood, and then they're activated by due to damage to your endothelium due to external factors. And they basically all end up encoding for prothrombinase complex, which is an enzyme that converts prothrombin, or also known as coagulation factor 2, into thrombin, as denoted by factor 2A. And this basically converts fibrinogen into fibrin, which is a soluble monomer, and then it converts it into an insoluble monomer that reinforces the clot, causing it to not melt away and become strong. And thrombin also encodes for factor 13, which is a stabilizer, as the fibrin join together, forming crossbits and causing the clot to become firm and stable. Factor 13 comes in and acts as a stabilizing factor. Yes. And then after that, as you know, once the clot is formed and it's stabilized, you need to dissolve it as it's basically creating a huge mass in the middle of a blood vessel, and that's not very good for flow. And this process is known as fibrinolysis, which is the removal of clots. And this process is basically initiated by the conversion of plasminogen into plasmin. However, as you know, when the wound is healing, we don't want the clot to just disappear and your blood begins flowing out of the injured area. So during that, tissue plasminogen and urokinase are activators of plasminogen into plasmin, and these are inhibited by plasminogen activator inhibitor 1 and 2. And these are upregulated or produced in higher amounts during inflammation, as they are in response to cytokines within the endothelial cells, and this results in them being created more, therefore inhibiting the conversion of plasminogen into plasmin. However, once the wound has healed, these are then the levels of the inhibitors are reduced, resulting in plasminogen being converted into plasmin, and then plasmin breaks down fibrin into its degradation products. Yeah. And then these products are often measured in tests to work out if the patient has any clotting problems. And then an example of these are basically the hemostatic function test. Usually you do a full blood count and examine the number of platelets, as if you have low platelet numbers, you're going to have slow coagulation times. In terms of a coagulation screening test, there's a few major key tests that are done, which are the prothrombin time, which is basically a test in which measures the extrinsic pathway of how long it takes for a clot to form via the extrinsic pathway. And then it's the activated partial thromboplastin time, which is a measure of the intrinsic pathway. And then we have thrombin time, which is how long it takes for fibrin to get converted into, fibrinogen to get converted into fibrin. And then also you have your D-dimer and fibrinogen levels, as if those are low, that also means that there is not enough of the precursor to form fibrin. As well as platelet function tests, you can measure how fast they form a clot. And if they're not working correctly, you'll have decreased times, and as well as measuring the amount of coagulation factors within the blood, as reductions in those could be an indicator of poor liver function or some type of deficiency, maybe vitamin K, which is required for 2, 7, 9, and 10. Then we have hemostatic disorders, which result in the inability of a person to have their blood clot, and signs may include purple discoloration that are not due to bruise, mucosal or joint bleeding, muscular hematomas, and increased bleeding following surgeries, as they don't have the clotting factors required to clot. Also to this, there's also thrombosis, which is the formation of clots within the circulation. And this can result in the blockage of blood vessels and emboli, which are basically blood clots that break off, and then they can lodge into other parts of the body, such as the lungs or the heart, causing a heart attack, or into the brain, causing a stroke. In terms of why these occur, the Charles triad is basically what a science, not a scientist. I think it was a scientist who came up with the three main factors that are extremely important to thrombus formation, which is endothelial injury, which increases procoagulation factors. And this is in the sense that you have increased inflammation in these areas, resulting in decreased formation of, you have increased plasminogen activator inhibitors, therefore you get reduced number of plasmin, therefore you have reduced breakdown of clots, therefore you have increased clot formation, right? And then you get abnormal blood flow, which results in more contact of platelets with walls, as well as increased coagulation factors within the blood. And that this just results in a higher likelihood of thrombosis occurring, and as a result of that, the higher chances of embolisms. Sweet. That's a wrap on my part.
We got one more, I believe.
So final presentation of today. So now we're going to talk about how cells or tissue in the body respond to injuries. So cells typically in a stable state, homeostasis, and they just do their function. However, if there was a change or stress, they can adapt to this change or even respond to this change. Changes can be classified into physiological changes or pathological changes. Physiological, they are normal stimuli such as hormones. However, pathological, they are the abnormal stimuli that causes stress or injuries and result in changed cell structure and function. For example, if there was an injury or cut. And this chart here shows how normal cell responds to stress or injury. So we have two stimuli. We have injury stimulus and stress. Usually, if there's stress, the cell adapt to this stress. However, if it was like too much and the cell cannot adapt to this stress, it will be treated as a cell injury. And then we have two pathways for cell injury. It can be severe cell injury or mild cell injury. If it was severe, it will undergo cell death. But if it was mild, it will be considered as a reversible injury, which we'll talk about in seconds. So if cells undergo too much stress, it will be considered as a cell injury, and it has 5 signs, exactly the same signs of inflammation. So we have redness, swelling, heat, pain, and loss of function. So if it was considered as a cell injury, so we have 2 types of injuries: reversible injuries and irreversible injuries. So that if the injury was mild, it can be considered as a reversible injury. So a reversible injury result in tissue regeneration or adaptation. That means that the cell will not go under necrosis or cell death. Regeneration means restoration of normal tissue. Adaptation means growth of adapted cells. However, irreversible injuries result in tissue repair. That means that the cell will undergo cell death, will undergo necrosis, and different cells will grow, which is called scar tissue. There are three factors affecting the response, like is it will it be reversible or irreversible? Will the cells be regenerated or will go will undergo cell death? The first factor is the amount of damage, the size and volume of this injury. Another factor is the type of tissue and the types of tissue classified based on their capacity to regenerate into three types. So we have labile tissue, which involves cells that continuously proliferate, for example, skin cells. Then we have the stable tissue, which involves cells that are infrequent in proliferation, but they can rapidly proliferate, for example, pancreas cells. Finally, the permanent tissue, which cannot be replicated or regenerated. They will just go, they will just undergo cell death. The last factor is the duration of injury. And this table here shows how these factors affect the response of this injury. So if the size and volume was small and the tissue was labile or stable and the duration was considered to be acute, then the response will be regenerating the cells or adaptation. It will be considered as reversible injury. However, if the size was large and type of tissue was permanent and the injury was chronic, then cells will be repaired. And finally, cell death. We went through this before in week 1, and we will went again through this next week in much depth. We have two pathways for cell death. We have apoptosis and necrosis. Apoptosis is a result of physiological processes, so it's completely normal. It's very systematic. It's very organized and controlled, and it starts with caspase activation. However, necrosis is a result of injury or pathological stimuli. So it's not regulated, and we can call it the abnormal due to accidents or premature cell death with inflammatory products. So it causes inflammation, and it might damage the surrounding tissue. It might be caused by mechanical causes, loss of ATP, mitochondrial loss. And that's it for today. Thank you so much.
Yeah, that about wraps us up. So thank you everyone for coming. I hope you guys learned something. See you next week.